Hello everyone, welcome back to another session of regeneration biology. So we will continue with the position control gene, that is part B. So far we have been learning how different gradients contribute to the organogenesis. Now we will learn how different position control genes contribute to the establishment of a given organ in a particular spot. So as you can see in this picture, this is a planaria that is cut either at head or tail, okay, as you like it. And there are conditions like you can see in one situation when wind is inhibited, you are preventing the wind, that is a wind downward arrow, you end up getting two heads on both sides, and when you upregulate wind you end up getting two tail on both the ends. So this is the typical morphology. So the position control genes are continually and regionally expressed in planarian muscle to control adult body plan maintenance and regeneration. So there are two things here. One thing is having its expression, second is retaining its expression. So one part assures that the expression ensures a given organ, but in other side you need to have the prevention of, the opposite end should have the prevention of any gene that is going to favor an unwanted structure. So it is just like if you want to become rich, you have to earn more money and you have to also have to prevent others from getting more money. If everyone has got as much money as you are not rich. Of course you are rich, but you are not relatively rich. So this is the concept of Marius signaling. Not only one signaling is favored, it also has to make sure that the same signaling do not happen in unwanted place through inhibitors. So you can see here a regeneration patterning phenotype. Left, the trunk fragments that is regenerating the heads and tails depending upon the situation. If you knock down wind, you get head in both the ends. If you upregulate wind, you get tail on both the ends. Inhibition of wind pathway results in posterior facing. Anterior facing head is normal, which is perfectly fine, but posterior facing head is not normal. So inhibition of negative wind pathway, that is which leads to the regeneration of anterior facing tail. Posterior facing tail is normal, anterior facing tail is abnormal. In the right side, what you are seeing is BMP pathway. That inhibition leads to dorsal appearance of ventral attributes. In the previous class we saw dorsalization or ventralization, this word we saw, like you can see in this picture. So the method used is different RNAi, whether to downregulate wind or upregulate wind. These are all done through RNA interference. You can see RNAi of BMP, you end up getting the cilia which is on the top. So ventral cilia, which is the planaria has for crawling on the surface. Now you have both ventral cilia as well as dorsal cilia. And here in this B, it is the staining. The panel B, it is the PCG expression domains, that is anterior up, posterior down, and different genes are there. SFRP is a negative regulator of wind signaling, and it's not important to get into the qualitative nature of these genes, but all you see from this picture is different genes are differentially distributed and they are all position control genes. So now this picture, this cartoon says that how different gradients of various molecules are distributed. The brightest spot shows strongest and the faintest shows least. So many of these genes, if you see within this eye region itself, their gradient disappears, whereas some of them starts faintly and then increases and then disappears, like NDL1, NDL3, one example. And several winds are there, so their expression and then disappearance. So same way posteriorly also, if you see some of them are strong, some of them are weak. You may wonder, some winds are there present both anterior, some winds are present posterior. But remember, presence or absence of wind is not the stringent criteria, but whether that wind is inhibited by the proper inhibitor. So that is what the real hallmark. So you have activator as well as inhibitor. So the abundance of one of them will decide that which will get the upper hand. Like you can see here, this beta-catenin will be least in the anterior region, because beta-catenin, if it is abundantly present, then it doesn't favor head formation. Means you want low beta-catenin and stabilized beta-catenin, by the way, not expression of beta-catenin, but the stabilized beta-catenin. Towards the posterior side, you have got abundant beta-catenin expression that is present, you can see here. Same way in the D panel, like the cartoon map of the PCG transcription across AP axis, and this is beta-catenin is the only one which you should follow seriously, and other genes are basically contributing to the stabilization of the beta-catenin. That is nothing but the effector of Wnt signaling which goes into the nucleus. If you see this dorsoventral axis in the right side, that is a BMP, cartoon of PCG expression. PCG stands for position control gene. Okay, domains on the DV-ML axis. DV means dorsoventral axis. ML means mediolateral axis. As you can see here in this picture, you can see the expression of mediolateral axis, something very interesting. The Wnt5 is expressed right in this boundary, as you can see here. And in the middle, you have got a slit expression. And BMP expression is strong in the dorsal region. And also in the mid-dorsal also you have got the expression of slit genes. And also the Wnt slit mutual inhibition, it is important for the establishment of mediolateral pattern. So the importance of showing these pictures and pattern is to show that every region has got an identity, every part of the body has got an identity. Credit goes to these position control genes. Like you can see here, BMP is gradient, dorsal highest, ventral the least, and the medial and lateral. So medial to lateral, that means this is the portion to the lateral, and you can see the expression of slit is inhibiting the Wnt, and that make sure that wherever there is a slit expression, it do not want Wnt signaling to turn on. Wnt may be there, it is capable of stabilizing beta-catenin, but it do not happen simply because the slit will antagonize the Wnt. So the repressors also as important as that of the activators. And there are a bunch of genes which is listed here. That is the different genes are there. NOTAM, which is an antagonist of Wnt signaling. And if you have an RNAi phenotype, this is the gene and this is the RNAi. So if you get anterior facing tail, so that means if you prevent the Wnt signaling inhibitor, that means you are activating the Wnt signaling. So if you activate Wnt signaling, you end up getting tail instead of head. So same way, Wnt itself, which is the ligand, you are having posterior facing head. So we don't need to go into each of this phenotype. You can go through it one by one. There are so many genes are there, and it will affect, some of them lead to two pharynx, etc. So different phenotypes come up. So accordingly you will know, oh, this gene is important in doing this particular phenotype. Like this list is continuing, and name of gene is not that important. However, you need to know that whether or not a given gene is an activator or an inhibitor of Wnt signaling accordingly, or BMP signaling accordingly. You will have an anterior-posterior axis, or you will have a dorsoventral axis. So adult positional information is harbored in planarian muscle. So far we have been telling PCGs are there, position control genes are there, but where are they located? Who is holding? What is their address? So we know the muscles are static cells. They are part of your kind of your skeleton. Your musculature is a fixed. They will, although they can move relatively based their contraction, but they are not moving from one place to another. So these muscle cells harbor, or they are deposited with position control proteins. So the cells expressing PCGs are indeed muscle. So muscle is doing the muscle specific role, but when a neoblast is coming, walking across, then the muscle which is expressing a position control gene will tell that neoblast, okay, this area is meant for this, this area is meant for this, you become this. So body wall muscles includes longitudinal, circular and diagonal fibers across anterior-posterior axis, dorsoventral and lateral axis, that is the circular, and then tangential, angle to angle. So they are the diagonal fibers. These are the main muscles. Each of them have got some or the other position control gene expressed. So the MyoD is required for the regeneration and homeostatic maintenance of longitudinal muscle fibers. So another gene, NKX1, is required for the regeneration and homeostatic maintenance of circular fibers. And inhibition of both MyoD and NKX1 together lead to a substantial loss of body wall fibers and reduced PCG. That is what we are interested in, reduced PCG expression before animal death. That means a muscle is a muscle only when it is having MyoD or NKX expression. If you try to, it's just like, your identity is on your face. I just completely take one slice off your face, which covers your nose, eyes, mouth, everything, and I made your face flat. What is your identity? There is no identity. If you want to recognize, you have to do DNA fingerprinting. Same way, you get rid of the muscle's identity by knocking down MyoD or NKX1. Automatically, the position control genes also will get affected. Means when you make a muscle a non-muscle or you reduce its identity, the position control gene also equally will be lost. So what you learn from here, the quality of the muscle which you are referring to decides what should be the quality of the PCG expressed in that muscle. These animals displayed patterning phenotype. That means they have difficulty in forming proper pattern, generating extra eyes because you don't know which is, if there are position control eyes, position control genes specific for eyes should be expressed in a given muscle. Now you got rid of the quality of the muscle. They are expressed everywhere here and there, and then eye also will formed here and there. So there is no specificity. Further supporting the model that muscle has an instructive role. It is telling instructive role in regulating the adult planarian body plan. So the muscles being static in nature, they are the best candidate to harbor this position control gene. Gene means position control protein. They are the one which gives the guidance to the migrating cells, migrating neoblast cells. So the muscle rapidly regenerates the pattern of positional information. So whenever there is a damage, muscle should restore immediately and start expressing the PCG for the migrating neoblast to form a lost organ in that spot. How positional information regenerates its pattern is a central problem in regeneration. How now if a part is disappeared, like head is chopped off, now you have is a trunk. Now where is muscle? There is no muscle. It has to grow from the blastema. So first line of formation has to be the muscle, not the organ, and this muscle will start expressing the PCGs, and these PCGs will guide the migrating neoblast which is coming from rest of the body or from the remaining trunk. The pattern of PCG expression in the muscle rapidly regenerates after injury. So the PCG expression is very critical in every muscle that is being formed in the newly forming blastema or the regenerating tissue structure. Beginning between 24 to 48 hours after amputation, a tail fragment expresses numerous anterior PCG. Remember, fragment is tail. So since the anterior portion is being removed, the leftover, that is the posterior part, now start expressing PCGs responsible for anterior identity. Why? Because these cells now will move into the anterior region, future anterior region. And it has got the posterior expresses, and it will start expressing numerous anterior PCG at the anterior facing wound because they are just waiting to jump into the lost region or lost part, just waiting for. And the posterior PCGs restrict their expression most posteriorly. So from the existing, say for example, planaria was, let us think that one planaria was 10 centimeter. I'm exaggerating, 10 centimeter, and you cut off right in the middle, one half is removed. So now the remaining posterior body fragment has got only 5 centimeter. Now this 5 centimeter, which is only midway and posterior, anterior and until the midway is lost. Now this anterior most part of this 5 centimeter remaining fragment will start expressing maximum of anterior specific position control gene, and the posterior specific position control gene will be extremely posterior. Otherwise, they can be somewhere in the midway, maybe somewhere in the midway or maybe upper 2/3, it can be there. Because when you consider a 10 centimeter worm, maybe some posterior most, some 3 centimeter also posterior only. But now you have only a 5 centimeter fragment. Then the posterior specific region or posterior specific position control gene will be expressed more posteriorly than what it was in the full-fledged animal when it was there. So then there is a relative. It is just like if you say if there is a lion and hyena, they will fight. That will be very serious fight. But there are no lions, then hyena will fight within themselves, right? So for knowing that who is the boss, something like that is happening in the case of this position control genes also. When a fragment is small, there itself they start distributing. This is my place, this is your place. So accordingly, the polarity is restored back with respect to PCG in the existing fragment, not in the regenerating fragment. Opposite process happens in an amputated head fragment. So now you are talking about the posterior fragment. About the anterior fragment, what happened? The posterior end will start expressing maximum posterior specific gene, and the anterior specific gene will be in the anterior most end of the anterior half. Like I told you, 10 centimeter, you cut into 2 pieces. The rapid regeneration of PCG expression domains as an upstream regulatory step is a central pillar of planarian regeneration. So the PCG expression is the backbone of the actual regenerative events. Wound signaling. Now we have seen in the previous class how the wounds are regenerating. The wound signaling promotes regeneration of positional information in the muscle. 2 key molecules, they are Wind and NOTAM. They are opposite. Wind is a pro Wind signaling, NOTAM is the anti-Wind signaling, are activated by wound signaling and contribute to the resetting of the positional information. One of the early steps of position control gene expression. Wind is activated generically at all wounds by 6 hours post injury. Wind is a sudden release of, like we have seen that sometimes the cells will rupture, like in the case of Hydra we have seen it, and they will release a burst of Wind ligand on the site. Wind RNA can cause posterior facing wounds to regenerate, ectopic heads or to tail to regenerate, indicating that Wind1 promotes posterior and inhibits anterior identity. That means head formation, Wind is not welcome. But wherever there is a wound, Wind is expressed. So someone has to control it. So irradiated, amputated head fragments lacking neoblast cannot make posterior pole, and yet they are capable of initiating the expression of some of the PCGs such as WNT-P2. This is one of the WNT related molecule. WNT-P2 expression starts. WNT RNAi head fragments, however, could not activate WNT-P2. Okay. Head fragments are unable to make it now after a amputation at the posterior facing wounds, and demonstrating the role of wound induced Wind1 and in a process of resetting the posterior positional information or identity. If Wind1 is generically activated means irrespective of the place of the location of the wound, irrespective of that, Wind1 is activated by wounding and promotes tail regeneration. How do anterior facing wounds regenerate heads? It's because anterior facing wounds should not have Wind, because Wind is not welcome for head formation. But how the head is being formed? It's a very interesting question, right? The answer is, at least in parts, it involves the expression of a gene called NOTAM, N-O-T-U-M. Whereas Wind1 is expressed in the posterior pole in uninjured animals, NOTAM is expressed oppositely. That means its expression is more in the anterior end, the anterior pole at the head tip, which antagonizes the Wind signaling. After amputation, unlike Wind1, NOTAM displays preferential expression. Wind, I told you, wherever there is a wound, Wind is expressed, but NOTAM is not like that. NOTAM has got some positional identity. Preferential expression at the anterior facing wounds. Anterior facing wounds, NOTAM, when it is expressed in a specific manner, it will antagonize the Wind. So doesn't matter whether Wind is expressed somewhere or not, NOTAM will make sure that Wind is not going to do its job. Hence, head can form. And NOTAM RNAi, if you get rid of NOTAM RNAi, animals regenerate tails in place of heads. That means Wind is activated. Wind is activated for no reason. wherever there is a wound. Means it is waiting for wind signaling to kickstart. But if NOTAM is not there to antagonize it, you end up getting a default tail and in the place of head, because for head to form, you don't want wind signaling. For tail to form, you want wind signaling. And the absence of NOTAM ensures there is wind signaling. Why? Wind is induced everywhere soon after injury, indicating that NOTAM promotes head and inhibits tail identity. That is why you have to knock down NOTAM, because NOTAM is a antagonist of wind signaling. So wind signaling at the wound site, wind and NOTAM are part of a generic wound response. Induction of wind1 and NOTAM results in a high wind signaling environment at the posterior facing wound, not in the anterior facing wound, because anterior facing wound you have got NOTAM, and wind inhibitory environment at the anterior facing wounds because of the presence of NOTAM, not because of the absence of wind. And that this promotes the initiation of the changes in the PCG expression that ensue. So the wind signaling is contributing because the wind along with the TCF go and bind to the DNA and turn on some gene expression, and these genes are nothing but PCGs, which are giving control to a tail formation or head formation. Beta-catenin is required for the maintaining posterior PCG pattern. Beta-catenin has to be stabilized for maintaining the posterior identity with the help of PCGs, respective PCGs, and preventing the anterior PCG pattern from being expressed in the tail. You don't want head in the tail region. So that is taken care by the maintenance of PCGs, which is facilitated by active wind signaling in the posterior end. An estimated 200 plus genes are transcriptionally activated within the first 12 hours following injury in planarians, regardless of the wound type. Doesn't matter what kind of wound you gave. Soon after injury, around 200 or more genes are turned on. That shows how important the position control genes are. The induced genes fall into specific categories such as those activated within 1 hour of injury, includes stress response genes and are expressed in most of the tissues, and genes in a second category are preferentially upregulated in a specific tissue. Some are generic gene expression events, stress response events. Some of them are upregulated only in some tissues. Like muscles can have the PCG expression in them. Muscle, epidermis and neoblast are some of these specialized tissues which express specifically some genes. Wound-induced genes, that means genes that are expressed because of presence of a wound within 12 hours post-amputation with asymmetric activation at anterior versus posterior facing wounds yielded only one dominant gene, which is NOTAM. So NOTAM plays a very pivotal role in determining the anterior identity of a planaria. Wound-induced positional information and re-establishment is required for its regeneration. The positional information or PCG's expression has to be restored as early as possible for the re-establishment of the lost tissue structure. One muscle wound induced gene, that is called follistatin, is required for the missing tissue response. Like in the previous class we saw, whenever there is a wound, immediately the neoblast will start proliferating. That doesn't happen when you just pluck off one eye. It will run the show with the existing neoblast. So how this is regulated? Who goes and tells that a tissue is damaged? So follistatin is required for eliciting the missing tissue response. Follistatin RNAi can result in failed regeneration. That means there is no monitor. It's like a supervisor. If there is no supervisor, you can't expect someone to do the job properly. So supervisor, although not doing the work, it makes sure that the supervisor makes sure that the work is done the way it should be done. So follistatin RNA result in failed regeneration. Follistatin RNA tail fragments fail to re-establish AP pattern. That means if a tail fragment, tail is already there, head should be formed, but the anterior-posterior pattern is lost because follistatin is not there. So supervisory is not there. You will not get this anterior-posterior pattern. AP pattern of PCG expression and accordingly never regenerate missing cell types if follistatin is knocked down. Instead, amputated fragments endure maintaining with turnover of tissues they do have for some time, because animal has to eat. If the pharynx is gone, brain is gone, it will not be able to make a living. But the existing pattern is restored. Means follistatin is not needed for running the show or maintaining as it is, but it is needed for regeneration to happen in planaria. Extracellular signal regulated kinase, also known as ERK, extracellular signal regulated kinase ERK, phosphorylation is triggered within minutes of injury. Soon after a cut, within minutes it will be turned on, and pharmacological inhibition of ERK activation blocks regeneration and multiple aspects of missing tissue response. Missing tissue response is there in follistatin. Follistatin governs. It tells the animal what has happened. If follistatin is blocked, supervisory is missing, you will not get a proper PCG expression. But same way, if ERK activation also is inhibited, you will not get neither PCG nor regeneration. So ERK signaling is required for wound-induced expression of many genes such as RUNT1, WNT1 and NOTAM. So ERK signaling decides where WNT should be expressed, where NOTAM should be expressed, where genes like RUNT should be expressed, indicating that ERK acts upstream of wound-induced gene expression, essential for regeneration. So these are all WNT, NOTAM, these are all wound-induced gene expression, but they are in turn regulated via ERK signaling. A failed regeneration in the presence of an inhibitor of ERK activation, alleviation of ERK inhibition does not allow regeneration to occur. So what you get, if you prevent the ERK activation, you don't get regeneration, but the alleviation of ERK inhibition, first you inhibited, then you de-inhibited, inhibition is removed, withdrawn the inhibition, that is called alleviation, does not allow the regeneration to occur. So ERK inhibition or ERK activation, these are controlled in a timely manner. So failed regeneration is the result in the presence of ERK inhibitor or ERK activation is prevented, you will not get proper regeneration. However, re-entry of such animals inducing a generic wound response. Say a wound was there, you block the ERK signaling and the wound is healed somehow, you have no regeneration. But if you want them to regenerate back, you have to make a fresh wound. That means wound response is must, induces regeneration, supporting a view that the generic wound response is important. Say for example, you have a finger, it is cut and a fragment is lost, it healed now. Now, if you want that finger to grow back, whatever cocktail of gene expression you know, you need to make a fresh wound. In planaria, a healed wound cannot regenerate. You need a fresh wound. Then only it can think of regeneration. In that situation, if you are allowing ERK signaling to go on, then you will get proper regeneration. So muscles is essential to memory. Wound-induced NOTAM and follistatin expression occur specifically in the longitudinal muscle fibers. Myotic RNAi led to progressive specific loss of longitudinal fibers. Consequently, NOTAM and follistatin were not induced at wounds in myotic RNA animals. If myotic is not there, muscle lost its identity and hence you will lose the expression of NOTAM and follistatin and these animals fail to regenerate. Myotic RNA animals like follistatin RNA animals were incapable of PCG re-establishment after amputation, and it was therefore proposed that these amputated animals could maintain whatever tissue they did have but failed to regenerate any missing tissue because they didn't know anything was missing. No one was there to monitor the missing part. These findings support the model that wound-induced PCG re-establishment is required for regeneration. So we will study more about this gene expression in the next class. Thank you.